Item: SCP-673. Object Class: Keter. Special Containment Procedures: no living tissue is to enter the main containment area while SCP-673 is present. Dual airlock systems separated by decontamination and scanning cells are to be maintained at all times. Containment cell is to be immediately locked down during any maintenance or power failures, with release only after review by site security. Equipment exiting the containment cell is to be sterilized and remain in quarantine for a minimum of 2 weeks before being reused. Containment cell maintenance is to take place only after SCP-673 has been removed to a temporary containment cell. All surfaces are to receive treatment by chemical agent Niox-9, with any overgrown areas extracted and rebuilt. Containment cell must be cleared of all personnel for no less than 12 hours before SCP-673 can be replaced to primary containment cell. Temporary containment cell is to be flooded with Niox-9 for no less than 48 hours after SCP-673 removal. Independent instances of SCP-673 are to be severed and isolated as soon as possible after initial discovery. Evaluation of newly severed instances is to take place within 5 to 8 hours post-severing. SCP-673 instances found to have no research, production, or other value are to be incinerated immediately after evaluation. Instances added to the main contained mass are to be recorded and logged with central records. Any staff working in or around the containment area are to be placed in quarantine immediately after completing their given tasks. Observation of quarantined subjects is to continue for a minimum of 2 weeks. Any erratic behavior, growths, or other SCP-673 symptoms exhibited by subjects is to be reported to Site Command, with subjects being held for eventual evaluation. Description: SCP-673 is a mass of organic tissues, connected in what appears to be a totally random assembly. These tissues include recognizable structures, such as hearts, digestive tracts, blood vessels, and brain matter. However, many tissue structures are not identifiable by composition or purpose. Tissue structures are also noted for their increased size, appearing 20 to 50 times as large as their normal counterparts. Sample tests have shown the basic structure of SCP-673 to have no known counterpart among normal organics, and lacks any form of DNA or normal cellular composition. Tissues appear to operate without the need for any outside stimulation or substance, with hearts beating for days with no blood or nerve connections, and lungs continuing to expand and deflate even when severed and fully submerged. Tissues, isolated from the main mass, will grow into surrounding structures, using what appear to be tendons or thin bone-like structures to penetrate and spread over walls, floors, and ceilings. Tissues will operate indefinitely, with no tissue decay normally associated with exposed organs. SCP-673 appears to be infectious by an unknown means, with physical contact being the only constantly observed transmission method. Instances of airborne transmission have been reported, but are extremely rare. SCP-673 appears to prefer organic tissue, but will integrate with inorganic structures at a slower rate. Testing has shown that tissue integration appears to progress along loose guidelines in regards to inorganic structures. Pipes become veins and tracts, beams become bone-like supports, doorways become valves, etc. in a slow progression. The Shell Project, too, has shown that, with sufficient space and ample amounts of organic materials, SCP-673 data expunged